So thank you very much for attending this exhibitor tutorial. We are pleased to talk to you, present you our data around a versatile hit finding strategy to address drug discovery programs. So let's start. So how can we approach hit finding? There is several approaches we can answer this question and find attractive hits to initiate successful hit-to-lead programs. First method is based on literature, basically by homology of the described molecules. We can run virtual screening and also run fragment-based screening and the classical, and I would say quite old technologies, the classical HTS screening. All of these methods are complementary, can be run individually or in parallel, and that require a robust screening cascade to combine them efficiently all together to get high quality data at the end. All those approaches can be for well-established targets or for entire novel targets. And HTS is definitely a proven method to identify new structural chemotype and to initiate successful programs. So today we will take you to two case studies around hit finding. The first one will be about our PIM3 kinase program, and the second one will be a case study around GPR 183 for the Crohn disease. So I will start first with the PIM3 kinase program. So PIM3 is a kinase, part of a family of three serine-threonine kinase, PIM1, PIM2, and PIM3. They have a very highly conservative kinase with a unique binding pocket for ATP, and they are constitutively active. It has been reported some overexpression of the PIM3 kinase in certain cancer and tumors. And so it makes both targets very attractive for cancer therapy. To initiate a hit finding program, it's important to get the right tools. For these specific programs, we develop a several number of technologies. We basically have a very comprehensive toolbox. Oh, sorry, mistake. We developed three approaches in this PIM3 project. So the first one, I will present you the data around a conventional HTS program, basically based on a classical HTS screening cascade combining primary assay and orthogonal assays. The second approach I will present you is the virtual screening approach. And the third one we will finish by the fragment-based screening approach. And now the toolbox. Sorry for this mistake. So as you saw, there is plenty of color corresponding to different assays. And all of the assays are combined within screening cascade. But it's important to have the right tools to be efficient and having good data. So for this project, we use our protein production quality. You need high quality protein for any biochemical approach. We also develop primary assay screening using the ADP-Glo, gold standard for HTS for kinases. We also develop mass spec activity-based assay as an orthogonal readout to confirm the activity of the hits. And we use the automated system, the rapid-fire mass spectrometry system. We also implement a cellular target engagement to validate the activity of the hits in a cellular context. In this case, it was the nanobread, that is a cellular binding assay. We also characterize the hits using biophysics method. So we develop assays in SPR and MST-TRIC, spectral shift, that give us KD values of the compounds, direct binding on the target. At the end, as you will see, we managed to investigate the selectivity of the hits for the kinase. I mentioned there is 3 kinases in the PIM family, and it's quite important if we can rank and prioritize the hits for their selectivity versus the other member of the family. So for that, we have access to our kinase platform, the kinase profiler and the kinome scan, to quickly investigate the selectivity of the hits. And finally, we also characterized the hits we got in early ANME assay, we could say, for the solubility and the hep stability. So I will start to show you by presenting you the data of the HTS approach. So for this first part of the program, we combined several assays, and we screened over a part of our library, in total 73,000 compounds, combining a diversity set and hinge binder kinase focus subset. For this HTS, we have a TPP, a target product profile, quite early at this stage, but we wanted active hits in the primary assay in ADP-Glo. We wanted the hit being confirmed in orthogonal assay, in the mass spec assay, and also confirmed in the binding assay, the biophysics approach. We wanted also these hits to be chemically tractable, so with the possibility to generate IP and innovative scaffold. We also wanted the compounds being active in a cellular context, so in this case in a nanobreath assay. And at the end, we wanted some selectivity, if possible, and also having some compounds with acceptable ANME parameters. Let's start with the ADP-Glo data. So if we look at the pilot screen, so it's a subset of compounds to validate the robustness and the repeatability of the ADP-Glo assay. Being sure we will be able to screen with a high quality the 73,000 compounds. So on this graph, I will detail this first. I will go into details in this first slide, and after I will go faster. But in this slide, you can see the SNR, so signal-noise ratio value per plate, also the Z prime per plate, average value. And you can see there is quite a robust and repeatable, very robust assay. And if we look at the activity of the activity of the compound between the two independent experiments we run to determine the activity of the compound, there is a very robust and repeatable data because we found the same inhibition activity of the compound in the two independent experiments. And if we look at the frequency of distribution of the activity of the compound, we got a Gaussian-like shape of the curve. So basically, no bias in the activity of the compounds and the HTS. So we got a robust data, repeatable, and no bias identified. So all the lights are green to continue the project. So we screened the 73,000 compounds. I won't go into details, but we kept exactly the same quality parameter with even a better Z prime of 0.86. And as you can see, the Gaussian distribution is still the same and really robust. And so from the 73,000 compounds, we selected, our medicinal chemist did a selection of 1,000 compounds for confirmation in dose response curve. So we confirmed the compound in 8 doses confirmation dose response curve, and we confirmed 94.6% of the compounds. We determined an IC50 for nearly all of the compounds tested and selected, with some value that were up to submicromolar activity, with a PIC50 of 7.72. So we were very happy, excited, and with some very interesting scaffold. So now, after this first part of the experiment, the most important is to confirm the activity of the hit we found. So the second set of experiments we ran were the mass spec and the nanobreath assay. Still the same, we ran the experiment in 2 independent experiments to show you the robustness and the repeatability of the assay. And as you can see, on the top for the mass spec, on the bottom for the nanobreath, the correlation of the data of the PIC50 values between the 2 independent experiments are very good. So we are quite confident in the 8 we found in these orthogonal assays. And we have been able to confirm 92% of the compound in the mass spectrometry activity assay. On this graph, it's a correlation between the PIC50 in ADP-Glo versus an IC50 in the mass spec assay. Most of the compounds are confirmed. What you can see is there is a light shift in the potency of the compound. That is acceptable because the assay is different, the readout is different. Also, if we look at the same representation for the nanobread, so confirmation rate is a bit lower because we confirm only 50% of the hits, but that's kind of normal with the cellular context because you have the compound that doesn't pass the membrane, and so they can't reach their target inside the cells. And as you can see, we kept the ranking of the compound with a shift in the potency of the compounds. But all of the compounds shifted basically following with the same delta. So we have very good hit in ADP-Glo, confirm in mass spec, half of them confirm in a cellular context. So basically, we validate lots of our requirements in the perfect hits. So our medicinal chemists did some triaging and selection of the hits. So starting from the 73,000 compound for the single dose screening, we reached at the end we got 6 cluster innovative compounds with structures that are not reported as being kinase inhibitor or PIM3 inhibitors, and this cluster representing 16 hits. So once we got these 6 clusters, we went further into their characterization. So the next step has been to characterize them in biophysics. And in my introductory slide, I mentioned SPR and MST-TRIC. In this case, PIM3 is not working in SPR. So, but thankfully, we had the MST-TRIC spectral shift, and that's worked very well, and we got very robust data. So keep in mind, for biophysics approach, it's always good to try different approaches at the beginning, at least to de-risk, and to be sure to pick up a right assay to monitor the binding of your molecule. So the MST-TRIC spectral shift assay is a brand new technology released last September, and that combined the classical MST technology with an analysis also of the shift in the fluorescent spectrum of the protein. So with this technology, so you can determine the direct binding of the molecule on the protein, and also are integrated some quality control. So you monitor at the same time the autofluorescence of the compound, and if that make any artifact with the fluorescence you are following. And also this graph, it's a quality control of the several measurements we do. And if you have a spread lines on the top part, it means you don't have robust data. So in this case, all the lines are superimposed, so we are quite confident, very confident in the quality of the data. And we tested the 16 hits we get from the 6 cluster using the MST-TRIC spectral shift. And we confirm all of them by binding with some affinity between 0.27 micromolar up to 36 micromolar. And what you can see on the graph on the right, it's basically the MST approach, the biophysics approach, give us the possibility also to have a more precise ranking in the affinity of the compound, whereas in activity they were quite close to each other. So we gain in details and in ranking of the compounds. So all the hits we identify, so confirm in biophysics, so I was quite hyper exciting confirming all of my compounds, all of the compounds. So after the MST-TRIC, we check several parameters. So first the selectivity and the early ADME. I won't take you to all of the details. I try to put this table as a summary. Don't specifically look at the numbers, or if you can, but just look at the color. So when it's green, it means everything is okay. Orange, it's acceptable and need improvements. When it's red, it needs a major improvement because it doesn't fit with the minimum requirement for hits. So on the top part, you got the pharmacological properties, so ADP-Glo activity, a rapid fire mass spec, nanobread, and the MST-TRIC. And as you can see, sorry, except for one series that is not active in a cellular context, all of the clusters have a very acceptable pharmacological properties. For the cluster 1, it's up to our medicinal chemist to make the compound probably pass the membrane and reach the target. We won't make the life of the chemist too easy. So if we look at the chemical properties, they are perfectly, I would say, chemically tractable and very promising. In terms of selectivity, over the 6 clusters, we got already 2 clusters that show quite a reasonable selectivity for PIM3 versus PIM1 and PIM2, but things I was not necessarily expecting at the end of the HTS. I'm very sorry. And in terms of early ANME properties, except the first cluster, we have a limit of solubility, probably why we don't see it properly in cellular context. All of the series are very stable and very soluble in PBS buffer. So with this classical HTS approach, we have been able to successfully identify and characterize 16 hits distributed in 6 clusters. We confirm them both in activity and binding assay. And we have hits that are very good starting point for a need-to-lead program with good chemical properties and already some selectivities. So after all of the data around the HTS, I will take you through the virtual screening approach. So starting from the 6 chemical series, our medicinal chemistry, our virtual screening team work on 5 of the 6 series, and they work on those 5 scaffolds to be able to generate and explore some new chemical series. We wanted to explore and expand the chemical space covered by the hits and potentially identify new series. So for that, based on the X-ray structure of the PIM1 and the PIM2, they built by using AlphaFold system the PIM3 structure because PIM3 hasn't been crystallized. And so with the AlphaFold, with the PIM1 and PIM2, they have been able to create virtually the binding pocket of the PIM3 kinase. So once they have the model of the PIM3, so as a first step, they run a ligand-based screening, virtual screening approach using. So the first step was to run the ligand-based virtual screening approach. For that, they screen up to 9 billion of compounds, of virtual compounds. And from the 9 million, they selected the top 10,000 top scorers. And these 10,000 top scorers have been then docked into the PIM3 kinase model that has been built using AlphaFold in the model of the PIM1 and the PIM2. They rank the compound based on the docking score and potentially on the potential PIM1, PIM2, and PIM3 selectivity. And they generate several series and pick up 10 compounds of each. And at the end of, starting from the 9 billion of compounds, they selected 60 compounds to be purchased and tested, still using the same toolbox that has been used for the HTS. So the first assays we used was the ADP-Glo. So over the 60 compounds that have been designed and selected, we managed to get 46 of them synthesized and tested, first in the ADP-Glo. And using the ADP-Glo, we managed to determine a PIC50 for 20 compounds over 20 compounds over the 46. So that gives us a good hit rate of 48%. That is a very good hit rate in terms of virtual screening. We also try to investigate the selectivity. For that, we use the Kinase Profiler platform. Unfortunately, we didn't get any clear selectivity as we got from the HTS, but that's quite, I would say, normal due to the high homology of the PIM3 kinase binding pocket. But keep in mind that anyway, for the kinase, it's better to get this close selectivity for the closely related, but it's really the impact, the binding of the activity of the compounds against the whole kinase that is really important to avoid any off-target effect. Unfortunately, the MST-TRIC binding, the MST-TRIC spectral shift data are not available today. They will probably be finalized next week, so. But I'm quite confident we will confirm at least the 22 compounds. So this virtual screening approach actually generated new scaffold in comparison with the one we find in the classical HTS approach, and we use the same toolbox. So it means in parallel of a conventional HTS approach, we can have this virtual screening approach. And then to finish my part, I will take you through the fragment-based screening. And not as a surprise, we combine the same technology, ADP-Glo, mass spec activity assay, and we use our binding platform, the kinome scan, to look at the binding of the molecule on the kinase. Also, what is ongoing, it's the biophysics, and as for the virtual screening, I don't have the data yet. But we screen a part of our fragment library at the time that was representing 1,300 fragments. That is now, library is now expanded to 3,300 fragments. And so I'm not a chemist, but from what time I chemist, the scaffold is quite different and interesting, with very interesting 3D structure for the fragments. Sorry. So you are now familiar with all the representation. On the top part, you got the correlation between the two independent experiments measuring the percentage of inhibition of the fragment in the ADP-Glo activity assay. And as you can see, the data are very robust. All the activity of the fragment are spread all along between 0 and 100%. And we tested the fragment at 300 micromolar, so even at its high concentration, we can still manage to have a robust activity assay. And we got a hit rate of 1.37%. And when we run the dose response curve of these top compounds, we basically confirm more than 96% of the compounds with some activity that are below 1 micromolar, but is quite unexpected for fragments as well. We run the same set of compounds in mass spec, and as you can see, the data are quite good. There is more variation around the background. It's just due to the fact we inject a high concentration of fragment into the mass spec, and the mass spectrometer doesn't really like much that. But the top compounds, we kept the same active compounds, and the top compounds are really correlated. And it's the same, we get a very high confirmation rate when we move for those response curve. We are nearly 95% of the compounds, with similar activity as well. And at the end, we look at the binding. So the kinome scan is measuring the direct the binding of the molecule. So we determine KD in this case, and not PIC50. And it's the same. Single dose and went very well, and we got a very good hit confirmation rate in those response curve, and with similar affinity between, in comparison with the PIC50 values. And if we look at the graph at the bottom, it's just a recap of the two PIC50 and the PKD value we obtain. I know we can't really compare KD and PIC50, but in this case, for this class of kinase, the data are really robust and correlated between the two technology, between the two approaches, binding and activity. And as you can see, most of the compounds are confirmed with the same affinity or activity between the three technologies. As I'm very curious, we anyway try some selectivity, and you have the PIM1, PIM2, and PIM3 there. And as expected, fragments are so small that basically it's really difficult for them to be selective at this stage. But anyway, even if I don't have the biophysics data to show you today that are the gold standard for fragment-based screening, you have also the possibility to use activity-based assays So basically adding 1,000 and 3,000 compounds to your set of compounds for HTS doesn't represent lots of plates, and you can actually get some new scaffold. So I took you through all of those data. I'm very sorry, there is lots of data, but don't worry, for a real project, we won't necessarily develop everything. It's just a matter of what you need, what your project require. So if we consider developing, starting a project with a classical HTS approach, it's important to have a good library at the beginning and to have the right tools set up at the beginning of the project. So in this case, starting to develop the primary assay, the ADP-Glo, the biophysics assay. Don't forget to try several readouts. And then having cellular target engagement, selectivity assay, and some early. It's a nice to have, early ADME, that is a nice to have, but could help to prioritize the hit, especially for kinases. And as a backup or in complementary, or that can be taken on its own, it's to plug to this standard screening cascade some fragment-based screening that can go straight in the single dose screening or later stage and go straight for the biophysics approach. Also, the virtual screening, it could help giving more diversity, and the same that could give you the possibility to identify compounds that could join the single dose screening or at the later stage for the biophysics, because it's a reasonable number of compounds usually coming out of the virtual screening. So the idea is to play with this cascade as with Legos, and you combine the things depending on the need of your project. So I will give the floor to Rajan.
Okay. Thank you, Celine. Hi, I'm Rajan Chaudhary. I'm a computational chemist at Eurofins Discovery. I'll briefly go through the overview of how we look at HTS in combination with virtual screening. Then I'll provide our results on HTS and virtual screening, mainly focused on computer-guided results and conclusions. So this is a conventional HTS workflow which starts with assay development and validation for the HTS screening. Then you run HTS screening using the in-house or available compound library. Once you get some initial hits, you confirm them through reconfirmation assays. You probably take 10% of those compounds. And here comes an interesting step where you have to triage those HTS hits and predict some ADME properties, selectivity, with some medicinal chemist input. And then you select your final compounds, mostly for resupply or resynthesis for dose response confirmation. Now, how does the virtual screening looks like in combination or in parallel? For virtual screening, again, you have to start with the assay development and validation, which is probably similar to the dose response confirmation from HTS. Now, the virtual screen starts with either structural information of your target, or you start with the known compounds or hits identified from the HTS screens. Now, you screen a very large virtual compound library, and from that you identify your initial list of virtual hits. Again, you have to triage those hits with ADME properties, selectivity, etc. And then you select some molecules for virtual hits that you want to try or test. You run the assays, and from that you get your initial hits. Now, the advantage of running this virtual screen along with HTS is that you can rapidly identify novel hits, as well as save cost and time. Also, you can leverage at this virtual triaging system, where if you have some compound data from HTS, you can incorporate that in a virtual screening process, or from virtual screening, you can identify some compounds to triage in the HTS series. Now, one of the key important step in both of these approaches is the compound library, and how are you screening the entire chemical space that gives you best way to diversify your project hits. So I'll quickly go through the compound library for HTS and virtual library for virtual screening. So here I'm showing GDP17 dataset, which is enumeration of all possible compounds up to 17 atoms, carbon, nitrogen, oxygen, sulfur, and halides. And that chemical space is represented with the black dots in the triangle. Conventionally, similarities has been based on physicochemical properties or 2D similarity, chemical similarity. But in terms of structure-based or binding of molecule to the receptor, we know shape is as much important. So here on one side, we have rod-like compounds, disc-like, and spear-like. The Eurofins HTS library in 2019 looks like something similar to the yellow dots. So we have good representation of rod-like molecules, enough representation of disc-like, but very few representation of spear-like. Now, the dots in blue shows Presswick library, which is mostly FDA-approved compounds. And you can see some of those compounds go into the spear-shaped-like area. So we have been intentionally exploring this additional compound space, and by running this shape similarity measurements, we are increasing the diversity in our chemical library. So the 2021 library is shown in the green color. Okay, so this is HTS library, which range in 100,000s of molecules. What about virtual library? Now, again, just a reminder, most of the time, the main goal is to identify multiple chemical series that has SAR potential. And what is better than enumerating all possible synthesizable compounds that are available for purchase? Now, in the last couple of years, we have been seeing ultra-large libraries from Enamin Real, Wuxi, something in 10 billions or 2 billions. Today, this number is almost 3 times larger than the one in the slide. So, and you also have some proprietary partner collections, and this is around 13 million compounds, which are readily available. You place order, within 2 weeks, you receive those molecules. How do you take leverage of this large chemical space? Now, most of the time, the 3 main categories are hit identification, identification of pharmacological tool, and lead identification. So for the first 2 steps, which is hit identification and POC or pharmacological tools, you can run virtual screening, structure-based approaches such as molecular docking, but those are still computationally expensive when you have to dock billions of compounds. The alternative way is a ligand-based method, where you can identify based on shape and pharmacophore, and it's much feasible approach to screen billions of compounds. The second step is, again, rapid hit expansion. Because you already have the chemical space enumerated, it's very efficient in identifying SAR-related series from that space and optimize those compounds for further hit expansion. Okay. So GPR183. GPR183 is a G protein coupled receptor, and it's mainly expressed on immune cells, dendritic cells, and B cells. Its relation to Crohn's disease and ulcerative colitis. Both of these diseases comes under inflammation-based bowel diseases, and one of the main the exact cause of these diseases is not known, but it is said that environmental factors affect the immune system, and because of that, there is an inflammation. Now, one of the well-known model is stromal cells, CD34 positive stromal cells. They generate oxysterols, which are something like 7-alpha-25 dihydroxycholesterol. Now, they have a very high picomolar affinity with GPR183. And so when the concentration of these oxysterols increases, it basically the lymphoid tissue inducer like ILC3s get attracted in the region. And in a steady state, when you have dendritic cells, it forms cryptopatch, which maintains the homeostasis of regular function. But in the inflamed region, when the concentration of both oxysterols increases and also in combination overexpression of GPR183 on these ILC3 cells, they generate cytokines and results in inflammation and epithelial cell damage. Also, the study shows that if you knock out GPR183, it can reduce ILC3 levels and improves the colitis score. So the hypothesis here is if we antagonize GPR183, we'll reduce ILC3-driven inflammation and therefore reduce the severity of IBDs. Okay, so when this project got started, we did not have any GPR183 structure available, and also there wasn't a good small molecules to start with. So we decided to start with a conventional HTS approach, identify hits, and use those hits to then run the virtual screening. As Celine gave detailed information about the various primary, secondary assays and HTS screen. I won't go into detail, but the HTS campaign looks like this. We screened 350,000 diversity set compounds in Lance cyclic AMP assay against human GPR183 in HEC 293 cells. From this HTS campaign, we got around 1,000 hits at greater than or equal to 90% inhibition. Now, these hits are then filtered out through reconfirmation assays such as IP1 and running selectivity against very key GPCRs. The number goes down to around 100 hits. These 100 hits then further reduced to 5 hits based on ADME predictions, discussions with medicinal chemists, identifying chemical series which is novel. And so these 5 hits were then input for the virtual screening. Now, by the time we got these five hits, there was a Cryo-EM structure of GPR183 published with a GSK compound. Now, it's a Cryo-EM structure, but the structural information still gives us ability to run both ligand-based as well as structure-based approaches. So here is a quick overview of our computational approach, which starts with large or ultra-large library for ligand-based approaches, where we use Bemis-Markov core selection as well as shape screening. And then in the second step, we do structure-based approach to filter, to generate additional virtual hits. These are then filtered down based on our in-house ADME filters, PAIN filters, CNS, different properties, including discussions with medicinal chemists. At the end of virtual screening, we purchase 100 compounds, and in the next slides, I'll give you results on each of those approaches. So the first one is ligand-based approach, where we take leverage of this massive cloud-based Orion platform, where they have generated, they have collected the 8 billion compounds. In the first step, we calculate similarities based on 3D similarities, and then cluster them according to the variety of scaffold. So from that clustering, we identify 10,000 top scorer compounds, and then we use these 10,000 compounds for our different approaches, ligand-based, shape-based, and docking. So for ligand-based, we generate Bemis-Markov core selection. We apply Bemis-Markov core selection approach, where you take a ligand, you replace heteroatoms with carbon atoms, you strip out the side chains, and it ends up with a very core scaffold of the molecule. Now, you can use some of the variations where you can keep heteroatoms in the ring system. That gives you more information about the chemistry, and which helps you selecting in different pattern space or different chemistry-wise. Once you identify these BMS cores, you filter them again with ADME predictions. And from this approach, we identified 25 compounds. We synthesized and received 23 compounds. Now, the very interesting part is we got around 18 compounds that had potency of less than 5 micromolar. Now, that's a 78% hit rate. This is one of our most highest hit rate that we have got. But in terms of 78 hit rate doesn't mean much. The most interesting result here is we were able to identify 4 compounds with less than 1 nanomolar potency. So that shows that we can explore very large chemical space and identify a potent molecule through this approach. So now, when we run this large ultra high throughput ligand screening, how can we leverage this data in another ways? So we have already got this 10,000 top scores. So we use those 10,000 compounds for shape-based screening. So here we are looking at 3D shape of the main compounds and then identify new compounds just based on shapes. So here again we identified 25 compounds. We received 18, and we got 7 compounds less than 5 micromolar. The hit rate is 38%. Now this is much lower hit rate, but it is useful because with shape-based approach you get the most diverse chemical hits, which gives you leverage on running newer chemistries. So what about structure-based approach? Now, for structure-based approach, we started with a Cryo-EM structure of resolution 2.98. Now, a lot of very rigorous effort goes into the structure-based modeling, where depending on, first you have to do quality check on the structure, loop modeling, as well as you have to make sure that that structure is correlating with the SAR series of each different chemical series. So we docked these 10,000 compounds into the GPR 183 Cryo-EM structure and selected around 10 molecules for each of the lead series that we had. So total of 50 compounds. From that, we received 33 compounds, and out of 33, 18 had potency less than 5 micromolar. Now, 55% hit rate is again a very good number. That number changes based on different receptors, but the major advantage of running structure-based approach is it provides you additional information about the molecule, where you can optimize ADME properties and other properties. Now, the difficulty comes when you have identified multiple chemical series from ligand-based approaches, and it's very difficult to dock those compounds into the single Cryo-EM structure. When we identified various compounds, it was very difficult to dock those compounds into the structure that we have, and therefore we hypothesized that there must be multiple conformations to this GPCR structure. So we did end up getting our own in-house Cryo-EM structure with a lead compound that we were working on, and to no surprise that there were very large conformational changes in the binding pocket. But getting these in-house structures now, it's helping us to optimize ADME properties. And so in conclusion, the ligand-based approach, when you are screening very large chemical space, it allowed us to identify potent molecules, and combination with structure-based approaches, we were able to rapidly do hit expansion as well as identification of diverse chemotypes. And seamless integration of HTS and computational methods, it really allows us to early selection of lead series with reduced ADME risk. Okay, that.
Yeah. Thanks, Rajan. And as a final slide, after all of this infos, so I hope you we took you through these two case studies, and we try to show you that the hit finding can be addressed by several approaches that could be complementary, and sometimes don't hesitate to relaunch a different approach at the end of the project to just rescue it just in case it could work. And for that, it's really important to have a very comprehensive and a robust screening cascade in place that is actually that has to be customized for each project. So combining primary, secondary assay, important to get biophysics data, to get the Crohn, K-off, and KD of your compounds. Also, if it's possible, to get cellular target engagement assay after all of this characterization, and combining this classical HTS approach with some fragment-based screening, and also with the virtual screening approach, machine learning, that could also help us for the early ANME parameters prediction to just save time to initiate the hit-to-lead program. So we took you through our vision of the hit finding. What we also noticed that is really important is to have a dedicated team to support the project. Project manager, it's important to keep the timelines, being sure everything is running smoothly, and combining all of the assays all together without wasting the time between the different phases. Scientific project leader, access to the scientific experts as well. And at Eurofins, we try to keep also constant innovation in terms of technology to really accelerate the drug discovery. I show you very quickly some data about the MST-TRIC spectral shift technology. We also invested in the ECO-MS, so high-throughput mass spectrometry that could accelerate the drug discovery. So it's important to keep an eye on that and to try to save the time, because time is money. So we really hope you enjoy this exhibitor tutorial. We have a booth, number 848, if you want to continue to discuss, or we still have a few minutes for questions. We also have two posters, one about the ECO-MS and one for the PIM3, if you want to continue also to discuss. Thank you very much, sorry, for being there. Yeah, we still have 10 minutes, 12.
Thank you for the presentation, it was fantastic. So the question is regarding when you have competing fragment-based conventional screening approaches in parallel, particularly the fragment-based screening. So the project receives molecules from the conventional screen before on the fragment, so that they focus on those molecules and they tend to ignore the second one. So in this case, did you synchronize the two for the outcome?
Yeah, in the activity-based assay, we try to screen actually in ADP-Glo the small molecule and the fragment library on the same time. So that was easy, and just several extra plates. And what is great is to get all the assays set up at the beginning of the project. I have in mind the biophysics. So if you have it nearly ready at the beginning of the conventional HTS, you can keep your chemist under control and release them the data for fragment and small molecule nearly on the same time. So if you have all tools ready at the beginning, you can manage their expectation and take them to wait, even if it's difficult.
No, we do see, because we are screening them from those 10,000 compounds coming from the ligand-based approach. Within shape-based and docking, sometimes we do see some overlap, but not much.
Yes? Yeah, so for the small molecule, we screen them at 10 micromolar for the single dose, starting 30 for the dose response curve, 30 micromolar, and 300 micromolar for the fragment. that has very weak compounds. They are weak molecules, so basically we screen range between 100 and 300, depending on the solubility of your fragment. In this case, we know they are soluble at 300 micromolar. I don't have it in mind, but it's really dependent on the KM of the enzymes. For PIM3, it's quite a low concentration. And after, you can play with the concentration of ATP to try to identify allosteric and competitive inhibitors. But it's at the KM anyway.
One more question regarding the MS activity-based assay and
yeah.
You showed correlation comparing that to the ADP-Glo assay, but I'm wondering in general, do the MS activity-based assays can be correlated with the assay for the compounds that were soluble?
The correlation between MS, yeah, the correlation is a bit more spread than with the ADP-Glo because we have a bit more variability in mass spec, but it's still really coherent. And I quickly show it in the table, but we are the potency of the compounds is kept between the different assays, whatever the readout. Yeah. But yeah, the cellular assay make some it disappear just of the membrane and the solubility.
And I have one last question regarding your cascade. I see you have a counter screen. Does the biophysical
Yeah.
or the mass spec activity-based assay take the place of the counter screen for the ADP-Glo assay?
What would be the, sorry, what?
For the cascade,
Yeah.
I see a counter screen just in case.
Oh, no, yeah. We didn't put any specific counter screen. As we have several different readouts to confirm the activity, we basically made the economy of of the interference assay, depending on your readout you got. With ADP-Glo, the number of false positive is quite limited. So, and we use the other readouts. Mass spec, no labeling. So basically, you don't have this kind of interference with fluorescence and luminescence. So that was a win-win situation, let's say. So as much as possible, if we can manage to get orthogonal assay that avoid a pure technical counter screen, we try to have it. But it's really dependent on the readout. Yes. Yes, on the poster 1310, or stop at the booth, we have some preprint, a print version of the poster. We can also discuss it if you want.
Yeah, that would make sense.
Yeah.
Do you have the paperwork for the slides that you showed at the booth?
We have some posters, yes, and we have a case study that's basically present most of the data as well. Yeah, no, we managed to resupply them from the library provider, so we had a fresh solubilization. So we confirmed them in those response curve. So. Yeah. I think we have to leave the floor for the next talk. Thank you very much.